Alright, so I will be doing a simulation of meiosis by using these paper chromosomes. Before we get into this specific view, there would have been interphase, which is how a cell spends most of its life in. As it goes into prophase, chromosomes would condense, and they would pair up. Two sister chromatids would form a chromosome, and they'd be attached by a centromere. In this case, for meiosis, we have two pairs of homologous chromosomes for each chromosome. There are 8 types, 7 autosomes, and 1 sex chromosome. As they would actually, in prophase, is also when they would cross over. So for example, if we're looking at this pair right here, two sister chromatids are attached by a centromere. This segment could cross over with this segment. It would have to be the exact same amount of genetic information. It would just be flipped over, so that a chunk of the moms would be on the dads and vice versa. Although I won't be doing that because that would involve ripping up these beautiful chromosomes. So now we'd go into metaphase 1, which is when all of these would align across the equator of the cell. This is when independent assortment happens, which is that there is no specific order or positioning for the chromosomes. The moms could all be on one side and the dads could all be on the other. Could be 50-50. You could have one of the moms and 7 of the dads on the same side. It's random, and this gives more genetic diversity. Let's do that. It's still visible. Let's put one on this side. Getting a bit dark over there. Alright, that's how they're going to be divided up for this model. From here in metaphase, there would be two centrosomes, one over here, one over here, from which spindle fiber would come out, and this is where anaphase 1 would start. The spindle fiber would attach to each chromosome on this side, and the ones coming from this side would attach to each of them on this side. They would pull them all over to this side. And these would get pulled over there, which splits the cell up in 2. And now in telophase, there's the actual separation, which is when cytoplasm starts to split up between the two. There's the start of a cleavage furrow. And then normally, if this was mitosis, we would go back to interphase. Although in meiosis, there are 2 rounds of it. So now we'd go straight into prophase number 2. And that's when the chromosomes would recondense, because they've decondensed during telophase. They'd come back together, they'd pair up. Now we're left with 2 sister chromatids, 1 chromosome of each. They would line up across the equator, yet again, for both cells. From here, centrosomes, one here for this cell, one here and one here for this cell, from which spindle fiber would come out of these. They'd attach to the sister chromatids in this case, and they would pull them apart. Same goes for this side. So this was anaphase. Now telophase. They would all clump up together, come back into the nucleic envelope. 1, 2, 3 and 4. Bam. These are now our new cells. So we've gone from 1 diploid cell to 4 haploid cells. This is meiosis.